In this biochemistry video, we'll be looking at nitrogen base. What are nitrogen base? Nitrogen base are nitrogen with organic molecule that usually forms the building block for the DNA and the RNA in our body. So nitrogen base, again, they are nitrogen that contains organic molecule that usually forms the fundamental building block for our DNA and RNA. So now, what are those nitrogen bases that we know? The nitrogen base, there are 5 of those. Nitrogen base. All right. And the 5 of those is number 1, we have the adenine. Number 2, we have the guanine. Number 3, we have the diamine. Number 4, we have the cytosine. Number 5, we have the uracil. Now, these are the 5 nitrogen bases that we have. Each of these nitrogen bases, the pair of those nitrogen bases depend on whether they are in the RNA or the DNA. Let's talk about the nitrogen bases that are found in the DNA. In the DNA, which is the deoxyribonucleic acid, the nitrogen base that is found there is the adenine, the guanine, the diamine, and the cytosine, AGTC. You can say that for yourself, AGTC. These are the nitrogen base that is found inside the DNA. And this nitrogen base need to pair one to one. They need to occur in pairs. How do they pair? The pairing here should be adenine pairs to diamine, guanine pairs to cytosine. So for each DNA, the nitrogen base will pair in form of adenine to diamine, adenine to diamine, and guanine to cytosine. That is how their base will be formed. Let's go to the DNA for RNA. Now, for the RNA, the nitrogen base that is found inside the RNA is known as the ARGO-C. Now, why do I call it ARGO-C? Now, so that I'll be able to note, or I'll be able to identify the base pairs. Now, the A pairs with uracil, and then the adenine pairs with uracil, guanine pairs with cytosine. If you are from West Africa, from the Igbo dialect, you can call it Ago-C. Ago-C. And what they mean by Ago is a lion. So this is just like someone's name, Ago. So it's Ago-C. So we can say Ago-C for that of RNA. If you're an Igbo guy, you remember it quickly. But if you're not from Igbo, that's not a problem. Just Ago-C. All right. Now this means that A will pair with U, adenine with uracil, guanine with cytosine. Now, up next, we need to know what is the structure of adenine, what's the structure of guanine, uracil, and purine and cytosine. Before we know that, we need to talk about the purine, and the pyrimidine. What are the purine? The purine, they are these nitrogen base that has a larger double structure. And then the pyrimidine, they are these nitrogen base that have a simpler structure. Now, the structures of the adenine, and the structure of the guanine is they have a structure that is double structure, a complex double structure. All right. Now, for the pyrimidine, which is the cytosine, the diamine, and then the uracil. They have a simple structure. Now, for you to remember this in a nutshell, to remember it very quickly, just write, let me make a space, write it here, AGTCU. Now, if you have done that, from here to here, they are known as the purine. So, this is the purine. Then, from this guy to this guy is called the pyrimidine. All right, you should be able to differentiate between the purine and the pyrimidine. Let's go to the next thing. The next thing is to draw the structures of all those guys there. What does the structure of the adenine looks like? Now, for the adenine, the structure looks like this. Something like this. With an amine, with a double bond. I hope you're following. And something like this. And then, like this. Then like this. Double bond here, and double bond here. So over here we have nitrogen should be here. Nitrogen should be here. All right. So we have the hydrogen. All right. So this is a structure of the adenine. What of the guanine? Guanine structure. Remember, the adenine and the guanine, they are the purine. They are the what? The purine. While the cytosine, diamine, uracil are the pyrimidine. So let's draw the structure of the guanine. Complex structure. They are double bond structures. Now, we have this. All right? So we can have a double bond here. We have amine here. We have nitrogen here. We have nitrogen here. So we have nitrogen. Then we have nitrogen. Okay, this is how the structure look like. Meanwhile, we have hydrogen here, all right? And then we have hydrogen here. Now, there's a structure of the guanine. What's of the cytosine? For the cytosine, AG, remember? My AGTC. All right, the next thing should be the diamine. For the diamine, I have a structure like this. Let me manage my board. All right, not that too artistic. All right. All right, so we have a double bond here. We have a double bond here. I have a CH3 here. We have nitrogen, nitrogen, hydrogen bond, hydrogen bond. Then if you can draw the diamine, you should be able to draw the remaining of those, which is the cytosine and uracil. They have almost the same structure, just a single structure. Let me manage the board over here. So I have this. Okay, nitrogen, having a double bond here, and then the amine. There is a difference between this is that this is replaced with amine, and there is no methyl in it. There's an alkyl or methyl. All right. Now let me make a space here. Let's draw the next one. We need to draw all of those. I have a double bond here. I have 9 nitrogen. I have a double bond here. Then we have a nitrogen here. So this is cytosine. This is uracil. All right, with this structure, we need to know that when you add nitrogen base plus phosphate group plus a pentose sugar, they're going to form a DNA, a nucleotide, which is used to form the DNA. So the nucleotide, contains the nitrogen base plus phosphate group plus pentose sugar. So these are what forms the DNA and the RNA. Now, if you look at the structure of the DNA, it looks like this. 5 sugar molecule. Now, this is not this is a RNA actually. RNA because there is no deoxygenation, no removal of oxygen from one of the hydrogen down here. Now, let me draw the DNA where one of the oxygen is lost. I need to use another pen entirely for that. Now, notice that over here, there's complete oxygen in the two of those hydroxide here. The oxygen is complete. But if you look over here, one of the hydroxyl has lost its oxygen. Okay, then we'll use my cleaner. Observe. Now, if you observe, you'll notice that one of the hydroxyl has lost the oxygen. That's why it's called the DNA, which is the deoxyribonucleic acid. If I add a phosphate group and a nitrogen base, and we have a complete DNA or an RNA. Now, let's find, okay, let's just use the structure. Now, if I should add a phosphate group here, all right, and then I decide to pick any one of these, Should we pick, let's pick something let's pick this. And then I decide to add it. Let's draw the structure of this one. We might not draw the structure of the next one. So let's just make a structure. What do you want to draw? I want to draw this. Then we have this. We have this. Okay. Now, this structure is what we refer to as the AMP. What we mean by AMP is adenosine monophosphate. So this is structure of adenosine monophosphate. If I should remove this, and replace it with, this is adenosine monophosphate. So if I should remove this and replace it with this, with the G, that will be guanosine monophosphate. If I do that over here with the DNA, maybe I put the diamine, okay, that will be what? You need to drop the answer in the comment section. If I should remove this one and put the C, which is cytosine, that will be what? Now, finally, if I should remove this and put the U, that will be what? So your assignment now is to go draw the structure of both RNA and DNA, having the base pair. Of course, it should have the phosphate group, having the base pair of we have not done this, diamine, cytosine, uracil. And you tell us the name and you drop it on the comment section. Thank you very much for watching. This is all we've got for you. Please have a great day. And in a subsequent video, we'll be talking about the nucleotide and nucleoside.